Hello friends, welcome to Divya Mahesh tutorial. Today we'll see DNA replication. So DNA replication means preparation of replica of DNA, parental DNA, like parental DNA, it will replicate and produce its replica. So this is known as replication. Various enzymes are involved and in stages. It occurs in stages. So firstly we'll see in DNA replication in prokaryotes. So for this we'll write here prokaryotic DNA replication. Before starting the process of replication, we should understand the enzymes involved in it and the DNA. So we know that double-stranded circular DNA in bacteria. We'll see the DNA application in bacteria. So it is double-stranded. So there should be a site from where the replication gets starts. So this is known as replicon. Replicon site where DNA replication starts, and also known as ORIC, origin of replication. So replicon is 245 base pairs. Replicon has 245 base pairs, and here this replicon is 80 rich bases. So in replicon where the replication starts, there 80 nucleotides are more in number. So that site is rich in 80. So now we'll see. So let us assume it like this. Rest of the part we should take it like this. So this is a DNA, rest is circular, we'll see later. So in this we will see ORIC, origin of replication. So it has 3 sets of 13-mer sequence. We have 3 sets of 13-mer sequences which are 80 rich, and from this DNA melts. From here DNA melts. So in DNA and ORIC region, ORIC, origin of replication, we have 3 sequences of 3 sets of 13-mer sequences which are rich in 80, and from here DNA melts. Now we have 4 sequences of 1, 2, 3 and 4 9-mer sequences. 9-mer sequences. We have 4 sets of 9-mer sequences for the binding of initiation complex. Here we'll see that from here DNA melts, and here initiation complex binds. So from here initiation complex binds. Initiation complex binds. So this is the ORIC, origin of replication. Now in DNA we'll also see there are for termination. Now this is for initiation and elongation from here. Now something there should be terminating. So there are termination sequences in DNA which are like this. Let's take this is circular DNA. So here These are termination sequences. So from here, termination sequences, one terminus contain ter A, D, E and other terminus contain ter B, ter C. Here also ter and ter. Ter A, D and E, and B and C. So these are termination sequences, and here termination utilization proteins, and they work in only one direction. So if we assume that this comes here, the daughter DNA comes here, so this will stop at here. This will, and when this will come, this will stop here. No one can stop each another. So one terminus at a time will be functional and only works in a unidirectional way. Now we saw sites where initiation complex will come and will sit over there and will start the process of elongation, initiation and all. Now we saw the termination sequences where the if whole DNA is formed, that daughter DNA is formed, then it will terminate over termination sequences. Now various enzymes are involved in it. So we will see DNA polymerases. As DNA is forming, so enzyme will be used as DNA polymerase. In prokaryotes, there are three types: 1, 2, and 3. When we'll see, this is the important enzyme which is used in polymerization, which actually polymerizes the DNA. But when we'll see the function of 1, we'll notice it can perform the function of 2, second, and also one more function, primer removal function 5' to 3', which DNA 3 lacks. So we'll take one by one these polymerases. So first we'll take DNA polymerase 3. DNA pol 3. DNA pol 3 is a multimeric enzyme made up of multimer subunits, various subunits. So we'll see each of them subunits. So it actually polymerizes the nucleotide sequences. So we'll take 2 alpha, we'll see tau, we'll see beta clams, and we'll also see over here more subunits. We'll name it, these are beta clams, beta clams, epsilon naught, these are epsilon naught units, these are theta units. We'll name it like this, you will be confused. So we will name it theta subunits, and also various gamma, delta, neta, and various subunits are attached. So this is the DNA polymerase 3. So we'll see these are beta clams. Beta clams, what it will do, that strands, they'll hold two of the strand and will slide, and where alpha, that is alpha subunit, will polymerize in direction of 5' to 3' polymerase activity. Alpha subunit is for 5' to 3' polymerase activity, and epsilon naught unit is for 3' to 5' exonuclease activity. So 3' to 5' exonuclease activity, that it will perform proof reading whether the bases attached are correct or not. So epsilon function is 5' to 3' exonuclease activity, and what is the function of tau is dimerization, and it get joined with the helicase enzyme which will unwind the DNA. So it will attach with this unit like this. If we take this helicase enzyme, so helicase enzyme is like this, it is hexameric ring, so it will attach there and it will start the process of unwinding. So we saw, and the function of theta is to increase their efficiency. Theta will increase the efficiency, and these subunits attached over the gamma, delta, meta over the beta clams, they help in loading beta clams. Loaders, they are actually called loaders. So they will load beta clam loader complex. So we saw alpha subunits which will actually polymerize in 5' to 3' direction. Then we saw the epsilon naught unit that will 3' to 5' exonuclease activity, that will perform the proof reading whether the bases attached made are correct or not. Then we will see tau, that is for dimerization and attachment of helicase enzyme. Then we will see theta subunits, that is for the maintaining the function of epsilon naught or and alpha, increasing efficiency of both of them. Beta clam, that will attach to the both the strands and slide over clam, like both will slide and the new DNA bases get attached, and these are clam loaders. So this was the DNA polymerase 3. So in DNA polymerase 3 we saw two functions. We'll see. We saw two functions. First was 5' to 3' polymerization, and second was 3' to 5' exonuclease activity, that is proof reading. So these two functions are of DNA polymerase 3. Now when we'll come with DNA polymerase 1, we will see, along with both functions, first 5' to 3' polymerization, second 3' to 5' proofreading. We'll write here proofreading exonuclease activity. Proofreading, it has an important function of 5' to 3' primer removal activity, that is 5' to exonuclease activity, that it will remove the primers. We'll see in the process of what are primers, we'll see in the process of replication, and we'll see the process stages over there what are primers. So DNA polymerase 1 has an additional function of primer removal. Although it performs all the three functions, but it's not the main enzyme because the efficiency of polymerization of DNA polymerase 1 is less than DNA polymerase 3. Efficiency of polymerization of DNA pol 1 is less than DNA polymerase 3. Now, and DNA polymerase 2, it helps DNA polymerase 3 and 1 to get into their function. So it has no such function for this. Now we saw DNA polymerase 3, 2 and 1. Now we'll see that this strand, it has to unwind. So for unwinding, we have another enzyme, helicase enzyme, that will unwind DNA. It is a hexameric ring. It is a hexameric ring and get attached to the tau subunit of DNA polymerase 3 and starts unwinding. It results in positive supercoiling of DNA. It results in positive supercoiling of DNA. Because of it, DNA supercoils, and thus another enzyme is recruited over there known as topoisomerase. Let's assume we have tooth rubber bands we'll have, and we make helix of that double stranded helix, and if we open at one side, so from both of the ends it gets coiled, more and more coiled. So because of this coiling, at a point when DNA helicase can't unwind the DNA, so there comes another enzyme, topoisomerase, which releases that coiling and makes the polymerase to slide easily. And how? There are two types, type 1 and type 2. It is single strand cut and it is double strand cut, and it uses ATP and no ATP over here. So in single strand cut, what it will do, like this, we will take here, we take this, let's assume this DNA is open, so it is coiled like this, and somewhere it meets here. Let's assume somewhere it meets here. So if we again and again will open here, will open. So at a time over here, somewhere here, will get like this coiling, and this creates problem. So here another enzyme is recruited called topoisomerase. So it will make single cut, either it will make a single cut and will release a tension over there. So tension will be removed. Tension will be removed and the DNA polymerase can easily polymerize the DNA. And in type 2, both the strands are cut and then tension is removed. Type 1 and type 2. Now next enzyme is primase. It is a modified RNA. So let us assume DNA is melted, helicase came, DNA melted at the AT rich site, and here is the initiation complex over here and here. Let's assume. Now what happens? DNA polymerase lacks the tendency to initiate the process of replication because we need some bases for the polymerization. These bases are called primer. So these primers are added by primase, RNA polymerase, modified RNA polymerase, which adds primer which adds primer to provide 3-OH free 3-OH group, so that another DNTP comes and attach with that phosphodiester bonds and again will provide 3-OH, 3-OH, 3-OH. So DNA replication starts from 5- to 3- position. So here we have all the enzymes over here in the DNA replication process. I hope you understood. So from the next lecture we'll start the actual process of DNA replication in prokaryotes with various stages, that are initiation, elongation and termination. Thank you.